Do you all see my screen? Okay. So just a quick recap of what we learned last time. So we were at the pair rule genes during early embryogenesis in fruit flies and their role in the pattern formation. And two examples we looked at were even-skipped and Fushi-Tarazu. So even-skipped is expressed in all the odd stripes, in all the odd parasegments, and Fushi-Tarazu is expressed in all the even segments. And the even-skipped means if this expression is missing, these parasegments, they get lost. And if they get lost, you have all the odd numbers gone, and you actually are having, just 7 parasegments. And that's why they say even skip. Okay. Now, yeah, we did talk about that the anterior margin of the stripe. So this is the anterior margin of even skipped here. This is the anterior margin of this parasegment, anterior margin of this parasegment, and this side is the posterior side. That actually corresponds to the compartmental boundary. That is the anterior boundary of the parasegment. Okay, so this is one parasegment, the anterior boundary of the parasegment, and then the parasegment goes all the way up to here. Okay. So the even skipped is expressed in nearly half of the parasegment. And the domains of other pair rule genes, because these are not the only 2 pair rule genes. There are other pair rule genes as well, which may cross the parasegmental boundary. Now we try to see how discrete gene expression of something like even skipped is established. In this particular case, we saw the second stripe, which is basically the parasegment 3 for even skipped. And what we learned was that, the least concentrations of giant and cruple, together with, a specific threshold of bicoid and hunchback, that leads to such a peak or discrete expression of even skipped. But at parasegment 5, it may be a different concentration of different genes, okay? Because here you have very high concentration of cruple. So it's not that always you will have least concentration of cruple and giant. And this is still a piece of puzzle how, different concentration, for example, here cruple is very high. So how then here even skipped will be established and maintained as well. But at least for the third one, parasegment, we do know what is the reason. We also looked at the promoter region of even skipped, and we saw that there are binding sites for bicoid, then hunchback, and then you have the cruple and giant. So all this basically highlights the coordinated action of activators or repressors, different transcription factors, which bind to the promoter region of even skipped or other pair rule genes. Now, the question for all of you is, there's an assignment question. You should go back and bring an answer to this question and submit it. So each of the cis regulatory site, for example, this is, each one of this is a cis regulatory site. And it controls expression in different stripes. How would you, your question is, how would you determine that each of this site has a role in gene expression of these payroll genes? For example, even skipped. So the question is, how are you going to experimentally determine, analyze that these sites have a role in expression or repression of even skipped? Okay. So today is Thursday, and you have till next Thursday to submit this assignment. Now, after payroll genes, as we learned that they are also transient, they disappear. And the gene expression of payroll genes is established by gap genes, which were established by maternal genes. So from the cascade of gene expression, which started with maternal genes, through gap genes, payroll genes, it eventually arrives at segmentation genes. And by the time we have activation of segmentation genes, and we saw engrailed is one of the segmentation gene, wingless is a segmentation gene, hedgehog is a segmentation gene. At this stage, embryo is cellularized. So it's no more syncytial blastoderm. If you remember, we don't have that photograph here. The earliest sign of segmentation are these 14 grooves, which indicate 14 parasegments. And what we learned that the payroll genes like even skip, they define the anterior boundary of each of the parasegment. Since the payroll genes are transient as well, now the question arises, how then the parasegmental anterior boundaries or parasegmental boundaries are fixed or maintained? If payroll genes are transient, transient means they are expressed in the anterior boundaries of the parasegments, and then they disappear. So if they disappear, who is going to maintain the function they initiated? How the parasegmental boundaries, they are maintained or fixed. And that's where the segmentation genes come in. So segmentation genes, they are not transcription factors. They are, unlike payroll genes, the segmentation genes are basically a diverse class of genes. They are cell signaling proteins. They are, cell signaling genes in this group of segmentation genes. So in this particular case, in segmentation genes, there's no obvious relationship to each other in their protein products. For example, here we saw, transcription factor bicoid is binding to promoter of even skip, etc. In case of segmentation, there is no such a thing, because they are not really transcription factors. They are quite diverse. And you need to have now signaling proteins, because this is the stage where you have individual cells. It's no more syncytial blastoderm. It's a cellular blastoderm, which means each cell is an independent entity, different than the neighboring cells. Now, the communication among these cells, there are many hundreds and thousands of cells like this. Now, communication among these cells, that is through the signaling proteins. One may be producing ligand, which travels to other. So through paracrine signaling or juxtacrine signaling, that is what is happening with the help of segmentation genes. So this is what we saw last time, the 14 parasegments. You see, and the earliest sign of these parasegments were, I told you, were just minor grooves. Sorry. These yeah. So these 14 parasegments are represented by these tiny grooves, and they coincide with the expression of pair rule genes. These pair rule genes, they also act in cellular environment because these parasegments, cells in these parasegments, they are now individualized. They are now, their fate is different. Had you been in cellular, in the syncytial blastoderm, then even-scaped and Fuchsia-Tarazu, they all will be freely diffusing in the cytoplasm and taken up by any cell in the nucleus of any cell. So one of the gene which we talked about last time was engrailed, and we learned that engrailed is expressed. Do you remember where engrailed is expressed? It's expressed in the anterior region of every parasegment. Now, last time we said if you have 14 parasegments, we said engrailed is going to express in the anterior region of each parasegment. And can you tell me what is the relationship between parasegment and segment? How do you define segment in flies?
So can I answer this?
Yeah.
Oh, so the parasegment, so basically the posterior of the first parasegment and the anterior of the next parasegment makes up the segment, and like that's how it goes on with the
Good.
continuation.
And if you have to now mark the posterior of an adult segment, what will you do?
Posterior, so we will use this engrailed, like we discussed in the last class, that you can stain it with the, do the immunostaining for the engrailed, so that the, because it's in the interior of the parasegment, so it will be ultimately present the posterior of the segment.
Good. I just wanted to make sure that you all remember this. So now let's look at how engrailed is being expressed and in relation to Fushitarazu or other pair rule genes like even-scaped, how it is present within the growing embryo. As I mentioned already, that engrailed is present throughout the embryonic segments, embryonic parasegments. Unlike payroll genes, which are in alternate stripes, so engrailed is present in posterior region of each of the parasegment throughout the embryo. It means if there are 7 stripes for even skipped, 7 for Fushitarazu, there will be how many stripes for engrailed?
14.
There will be 14 because engrailed will, well, when you will use immunostaining, you will see 14 parasegments. So this is engrailed expression, this one. Okay. Now, this cartoon shows you the expression of Fushitarazu, even skipped and engrailed. And so this one, the blue one, all this one is Fushitarazu. And magenta one are even skipped. Now, within the nucleus, if you pay attention, whether you have even skipped parasegment or you have Fushitarazu or you have even skipped, you have this blue dot which is engrailed. So engrailed is present in each parasegment, unlike the payroll genes. And engrailed is a segmentation gene, which is going to now define each segment, the posterior part of each parasegment. Now the interior region of the parasegment is engrailed expressing region. Which one is interior? So if this is one parasegment from here all the way to here, so the one which expresses engrailed, this one is engrailed expressing. This one is engrailed expressing. This one is. So the one which is expressing engrailed, this one, that actually defines the compartmental boundary, or we call this cell lineage restriction. There's no physical barrier here, you see. There's no physical barrier here. I'm just drawing a line so that you can see the interior compartment of a parasegment. There's no physical barrier, but there is a defined cell lineage restriction. Cells from here on are not going to mix with each other. This is what I was referring to about an ayat of Quran last time, that they flow next to each other, but they never mix with each other. There's no barrier here. There's no restriction, physical restriction, but there is a hidden principle or a mechanism which does not allow from here on cells of this lineage to be restricted with cells of this lineage. They will divide independently. They will divide independently. They will live together in the same organ. They will live together, contributing to development of same tissue and organ, yet they don't mix up. You will have, for example, if you see adult wing and you try to draw this cell lineage restriction based on engraved, you will see such a very clear line. Cells from this side did not mix with this or vice versa. So yeah, I explained you that the cells and their descendants from parasegment never move into adjacent form. Descendants of this parasegmental cell is never going to move this way or vice versa. And these domains of lineage restrictions, I was already using this word, is called compartment. They are restricted in one compartment. They're going to contribute to same tissue or organ, but they are never going to mix with each other. So now you can also do experiments by injecting fluorescent compound in embryo. And then that fluorescent compound can be activated by UV, very targeted activation of those fluorescent compounds by hitting a single cell with UV. And then you can trace that particular cell, because the one where fluorescent compound will be activated, when that will start shining under microscope, and you can see where it is going after cell division. We saw last time this drawing in which we saw parasegments, 14 parasegments, 1 all the way to 14. And we saw anterior and posterior compartment in each parasegment. And then we learned that posterior of one parasegment and anterior of next parasegment, that defines one adult segment. Posterior of one parasegment and anterior of next defines the next segment. And this segmentation pattern, it is fully conserved, or it remained preserved all the way to adulthood as well. So if this is head, C1, C2, C3, T1, T2, T3, so this is T1, T2, and third leg. This is third leg, the T3 segment. Okay, this is second pair of leg is in the T2, and this is first pair is in the T1. Then you have these A1 to A8, and here the abdominal segments are A1 to A8. Now we know this embryo, this is less than 12 hours old embryo. Already the segmentation pattern is established, and this is going to be preserved. Now, how this is preserved? The segmentation genes, engraved, wingless, hedgehog, etc. We are soon going to see examples of hedgehog and wingless as well, which are key segmentation genes. They play a role. We were talking about compartmental boundaries, and these lineage restriction or compartmental boundaries. You can do an experiment by using genetic tricks and try to see if you can disturb the cell lineage restriction, and what happens if the cell lineage restriction is disturbed. Or to begin with, you can ask a question, if it is really true that there is a cell lineage restriction. And this experiment is what we call genetic mosaics. Uh, and genetic mosaics are basically, you use in older times, they used to use X-rays. X-rays are going to produce mutation. And this mutation, because these cells are mitotically dividing, these are not meiotically dividing cells. These are now mitotically dividing cells. After mutation, when mitosis takes place, and if there is a recombination in mitosis, the chromosome which got mutation, let's say these two crosses are the ones which cause mutation. So after mitotic recombination, they will go, the two mitotic chromosomes, they will end up in the mutation will end up in the same cell, and the other daughter cell will be normal. Okay. Now what has happened is you have, this was a normal state of heterozygous, what do you call, heterozygous mutant, okay, carrying a mutation, wild type, mutation, wild type, mutation, wild type. So this is a heterozygous, okay. You hit with X-ray, X-ray is going to cause DNA damage. This DNA damage will be, the cell will try to repair. There will be a recombination induced during mitosis. As a result of that recombination, you will end up generating a cell which will be now homozygous for mutation. Both the homologs will carry the mutation instead of being heterozygous, which was the case here. Now, these homozygous cells, they will further divide because this is mitosis growth phase. And eventually what you see, you have a single bristle in the wild type due to, this is not wild type, heterozygous case, a single bristle which is just like wild type. And then due to mitotic recombination, due to homozygous mutation going into the progeny of single cell, you start seeing the phenotype. And then if you look at the adult wing, you have these bristles right here in the middle, which are normally only present at the wing margins. So what you have done, you have seen that the cell lineage restriction is disturbed. So this gene was controlling the cell lineage restriction, and now these bristles are appearing on the wing blade as well. They have done this experiment using engrailed, which proved that engrailed is indeed a cell lineage restriction gene. If you disturb, if you lose engrailed expression, there's no more boundary. You can see engrailed is mutated using genetic tricks, similar trick. And this compartmental boundary, which is very uniform in the wild type wing, is disturbed. Now, my question to all of you is, the second question you have to do is to use flip FRT system and induce engrailed mitotic clones to show disturbance in compartment boundary in fly wings? This is the question. You have to go back, read about what is flip FRT system, use flip FRT system, induce mitotic clones for engrailed, and draw a figure, draw a photograph how the compartmental boundary will look like. When you are going to induce flip FRT system, using flip FRT system mitotic clones, you will see a problem that your clones do not grow. Your clones are outnumbered. So let's say you induce mitotic recombination, you have homozygous cells. Now, there are, as a result of mitotic recombination, you have one like this daughter cells as well, which are so you, before you induce mitotic clone, you were like heterozygous. You started inducing mitotic clones, you have homozygous mutation and homozygous wild type as a result of. Now, the problem you see is these clones, they are not able to compete with wild type cells after cell division. The wild type cells, they outnumber, because they are, wild type cells are much more faster in cell division. Engrailed is a key gene, so we cannot afford to lose engrailed, because they are defective in the cell division, and they are outnumbered by these cells, and we call these cells, the twin clones. They are twin of these cells, and it's mosaic because you have heterozygous, you have homozygous mutation, you have wild type. Now, in order to overcome this, you use my new technique. And I would like you, when you bring an answer for this question, you should incorporate my new technique within your flip FRT clone. Is it clear? Hello?
Yes, sir.
Okay. So let's take a break here, and then we move on to segmentation genes again. Okay? Any questions so far? So if there are no more questions, let's take 10 minutes break. We see each other in 10 minutes.